thought this was an interesting opportunity to talk a little bit about what I used to do and still do. So like a story that essentially spans almost 10 years from when I was a graduate student all the way up to what my lab currently works on. An homage to the past, and then ending up with like, what do we do about the problem now? Because I think everyone can appreciate that inflation is real and it's getting really hard to keep science funded, especially in the last like 2.5 months or so. So let's just go ahead and get started. So the first thing, oh, sorry, this laser pointer, I just have to click back in the screen. Okay. So the first thing you might be wondering is like, what the heck are natural products? And so these things have a lot of different meanings to different people. So I think it's always good to think about like, what am I actually attempting to talk about here? So I think one common thing that you might have found in a store, botanicals and supplements are often considered natural products, but it can also mean very specific molecules that do very specific things. And these things are found everywhere. So plants make natural products, microbes in the soil make natural products. That's actually the source of a lot of our current antibiotics. You know, we can find natural products in the ocean, which is what I did when I was a graduate student here. And within the last 10 to 15 years, it's really appreciated that microbes in our guts and in and on our skin are also making natural products.
Well, you could have gone.
Oh. And the natural products, I was like, okay. These natural products are really, what it boils down to are specific chemical entities. So I've heard you learned about the central dogma, you know, when we think about DNA, RNA, and proteins. So I like to think about there's like one extra thing in the dogma, which is that proteins make small molecules, and then the small molecules can either be used to impact further gene regulation, further protein regulation, or just act as, you know, how does something over here talk to something over there? So we can think about these small molecules like pheromones, and, you know, how does one, you know, like how do the monarch butterflies always like go back to the exact same tree in Mexico? Like that's wild, right? I think a lot of these things can boil down to things that look like this. And these are somewhat related to proteins that you may have learned about. So they have, you know, we can look at these and like they may look really complicated, but, you know, we can see things that look like peptides, like this might look like a proline. It's joined here by an amide bond. But what we like about these, there's carbon, nitrogen, oxygen, and hydrogen. They have a lot of what we call stereochemistry. So that's the actual handedness. So I think one of the cool things about natural products are you can almost envision them as like keys that fit really nicely into proteins. So not every key is going to open every door, right? And the handedness of these molecules is like the bumps that give the key the specificity for the door. So we love these things, and a lot of these become drugs that we have used as anti-cancer agents, antibiotics, antifungals. You know, if anyone takes the statins, those were inspired by fungal natural products. So these things have a lot of uses, but the question is, how do we discover them? Because we know that they're in all of these environments, and microbes and plants everywhere have the capacity to produce these things, but they're actually really hard to find. And then when you find one, you're like, what the heck might it do? Because just looking at this, it, you know, if you can predict what this actually should be doing in the environment, I'd be really impressed, because that's the hard part. So I kind of want to tell you now a little bit vignette of like why we like natural products and how we think about them in my lab. So this is the structure of volinomycin. So this is,
there was a question.
oh yes.
How are they synthesized?
By enzymes that bacteria make, like large mega Dalton enzyme machinery. Like bacteria can dedicate up to 10 to 20% of their entire genome to make something of this size.
Using the usual standard model process.
Yes, yeah, so it's encoded in the DNA, and it'll read out to make the proteins to make this thing.
Right. It's probably worth saying it might take 25 different enzymatic reactions,
yes,
piece by piece, put them together
to make one thing that is less than, you know, Yeah, these are about 1,000 Daltons, so they're very small compared to the amount of like the elements and enzymes you need to put them together. It's crazy. So this is the structure of valinomycin. So this was originally shown to be a pretty potent antibiotic. So we know like its biological activity, like the nitty gritty is that it impacts potassium. So it'll help essentially throw off membrane potentials. And we know that this was actually, so, you know, it's cool that we found it, or that it was found as an antibiotic. And what people have later found is that it's actually produced by a specific bacteria that lives on the skin of a leaf cutter ant to protect it from fungus. And so I think this is an example of where these compounds have really cool therapeutic potential that we hope to exploit, but there's also a lot of applications in the native environment. And my other favorite example that I like to show is this structure here. So this compound was actually the first marine natural product to be put into human clinical trials as an anti-cancer agent in the 80s. And what we know about this, this is didemnin B. I was like, I haven't had enough coffee this morning, I think. So we know, you know, it was really exploited for a long time before its clinical trials were discontinued because it was too toxic. But they also found that, so it was produced by a marine tunicate. And what we later found was that if this compound is present in the tunicate, fish will not eat it. And then even further later, they actually found that the tunicate doesn't make it. It's a bacteria that lives in the tunicate. So there's a lot of these really complicated relationships. Like, so I think basically the tunicate probably gives the bacteria a nice home. And in turn, the bacteria provides a compound to help against predation from fish. And again, we have exploited these toxic compounds as anti-cancer agents. So I think these things hold a lot of really cool potential, again, as therapeutics. And then it's fascinating to think about how are these things actually used in their native environments. And so at the end of the day, when I think about natural products, I was told a long time ago that this is a field for people who love Sudoku and baking. Because we're oftentimes trying to piece together puzzles for like, what is the structure of what we're working on? What might it be doing? Because again, when we find these, we have no idea what they might be doing. So that's where we need to do Sudoku. And then for baking, I feel like we're oftentimes kind of like, how do we wing this to figure out what we need to do? So I hope that after I tell you a couple of vignettes, you'll understand why I think this is a pretty good representation. So we'll start with a little vignette that spanned about 10 years, more or less. So let me just say okay to this. Okay. And then we'll get into some more current stuff. Any questions before I go on? Okay. So this story actually starts with Pseudomonas aeruginosa. So if you're not familiar with this, this is one of the bacteria that people are most worried about in hospitals. Because this is what we like to call an opportunistic pathogen, meaning that like anyone that's in, you know, just like normally walking around would not be infected by this, but it's really bad for burn victims, cystic fibrosis patients, and anyone that's immunocompromised. And in burn victims, if Pseudomonas colonizes the skin, your skin will actually start to turn green. And that's how they know that that's bad. And we don't like this bacteria because it has become resistant to a lot of drugs. And so this is really the bane of hospital-acquired infections. So it's really important that we find, you know, we continue to find new drugs to treat these like bad bacteria. And the other thing you need to know about Pseudomonas is that it makes what we call biofilms. And this is part of why it's become difficult to treat. So basically, instead of thinking about it as a free-floating cell, Pseudomonas is really good at finding surfaces and building up a little house for itself. So it makes what we call exopolysaccharide, starts making extracellular DNA and extra proteins. And this picture is essentially, it has encased itself in all of this. And once it's like this, it's really hard for antibiotics to get in, or if they can get in, it's really good at pumping them back out. So biofilms are, again, like anyone with indwelling medical devices, this is what they're always worried about and testing for, because they do not want any bacteria on these surfaces, and especially any sort of like plastic surface is really susceptible to this. Another example of biofilms, like plaque on your teeth, is a biofilm if we're not, brushing regularly enough. Sometimes the scud on a shower curtain, like that's a biofilm. Biofilms are everywhere, but we don't want them in our bodies, and we don't want them on indwelling medical devices. And so this is, again, what Pseudomonas is really good at. And interestingly, there are no biofilm inhibitors on the market in the US. So if you have a biofilm, we really do not have a good way of treating it. So you can envision why this is really problematic then in a hospital setting. Okay, so
I
when,
have a question.
yes.
So, and that's like where it says dispersion, is there some pressure that builds up that causes it to burst?
Yes, so basically these cells can keep sloughing off and then essentially spreading biofilms.
Reinfected.
Yeah,
So not so much that they build up pressure, but that their biofilm gets burst somehow.
yeah, it's just, there's probably a limit for like how they can physically grow, and then at that point, it's like, we're a little bit too big, so we have to just keep like
Pushing.
pushing.
Okay.
Yeah.
Just one quick question. You said not on the market in the US. Are you saying that there are inhibitors in Europe, China?
I have not checked recently, but maybe at this point, maybe. Yeah. Yes.
So Laura, are these on surface? I mean, just surface removal, soap isn't a good agent to remove it, or alcohol or something like that?
Yes, soap is really good. But again, like if you have an indwelling medical device, you can't, soap it. But yeah, like for our hands and, teeth, again, you have like the physical brushing. Like they were trying to put biofilm inhibitors in toothpaste, because then you're not actually supposed to eat it, you would actually spit it out. So like the rules for like, what is tolerable in the application is a little bit flexible. But yeah, soap for surfaces is helpful, or just bleach if you can bleach a shower curtain. But you can't just, we don't want to drink bleach. I know that was advised a couple years ago. Yeah, that's a no-no. So yeah, so we want to think about how can we use natural products to maybe get rid of this, especially considering, again, if you think about like the marine environment, biofilms do grow on rocks. Like if you've ever seen a stream, right, we think about these bacteria growing all sorts of places in the natural environment. So this might be a good place to look for new drugs that might impact biofilm formation. So that's exactly what I did when I was a graduate student. So my former advisor let me do all sorts of weird things. And at the time, microbiome studies in the gut were really taking off. And I was intrigued because I was like, we are always collecting sand from the marine environment or sponges, but I was like, what about the things that like eat everything else? Again, thinking about the fish that, eat tunicates. And so I was like, I'd like to go buy freshly dead fish from Moss Landing, because then I didn't kill it and catch it, because then that would have needed extra permits. So I bought freshly dead fish and then cultivated microorganisms from their guts to see if we could find biofilm inhibitors. And then I had a reputation for about a year at a conference of being the dead fish person, which is fun, memorable still to this day. But basically, it kind of worked. So we found a bunch of really pretty looking bacteria, very different from anything that we normally found in sediment. So I do think animal intestines can be good sources to find new microbes that have different properties. And we did exactly what we set out to do. So we found a biofilm inhibitor called taurocholic acid. And what you're looking at here is actually an image of Vibrio cholera biofilms. So we did this in collaboration with Fitnat Yildiz, who's in microbiology just across the way. So again, I told you earlier that these things grow up into large 3D structures. This is what it looks like when you have Vibrio cholera that has a tag on it so that we can visualize it as green. And you're looking at it essentially from a top down. So these almost look like forests, right? So you can envision these large, on the Y and the X axis, these are really large 3D structures this bacteria is capable of forming. And then when we add our inhibitor at different time points, you can see that, especially in this image after 24 hours, that these large 3D clumps are essentially gone. So the bacteria is still alive and healthy. So we would hope to treat it with an antibiotic at this point, because it's no longer forming those large encasements of like sugar houses. So we're really excited. And then the other thing, so this is in cholera. And what we wanted to know is, did this work in Pseudomonas? And the answer was still yes. So again, we can see this is what the Pseudomonas biofilms look like. So we have these large 3D clumps that build up. And when we add our inhibitor, we don't see any of those large clumps, but the bacteria is still alive and healthy. So we were super excited. And that was basically when I graduated, and then I went off and did a bunch of other things. And then I was inspired to continue to work on this, because one thing that I'll share about these compounds, so this is taurourethylcholic acid, are that these are actually biofilm inhibitors, but they're also molecules that we make. So bacteria can definitely make these things. And for us, these are what our body uses to help digest dietary fats. So after, if anyone had like a croissant this morning, your body secreted a large concentration of these things to help you digest those dietary fats. And their regulation within our bodies is very tightly regulated. So you would think that like, okay, if that's like, you know, being secreted after I eat a meal, then I'm probably excreting a bunch of it later, you know, the next day. And that's actually not true. Your body recovers quite a bit of these. So you only lose about 1% of bile acids a day, because they're so tightly regulated. And basically, yeah, so what we knew then at this point was we could not use these as drugs, but could they be used as a tool to figure out like, what is it about these things that cause this effect? And originally we thought maybe it was just a simple, like, are they acting like soap? And we tested soap molecules and they did not work as well. So we do actually think that they have a very specific mechanism by which they're working. So even though these couldn't be drugs, we could still use them as tools to figure out what should we be drugging in this pathway. Okay, and so to start this, what we wanted to know first was, what were they doing on solid surfaces? So those images I showed you were at the air-liquid interface. But again, I kind of alluded to the fact that with Pseudomonas, you really want to know what's happening on surfaces, because that, I think, is when it's slightly more problematic. So we collaborated with Lars Dietrich, who's at the University of Columbia, and his student at the time, Lisa. And Lars is an expert in interpreting these patterns. So I think these are Pseudomonas aeruginosa colonies that are stained in red so that we can see what their biofilm looks like. And again, you could think about, instead of like these wrinkles are those large structures that these bacteria are so good at forming that we really don't like. And his website, if anyone is like further interested, I think these things are almost like art patterns. And they post a bunch of their really cool ones on their website. So if anyone is like, wow, I love this, he has all sorts of images, which is kind of fun, in addition to it being very sciencey. So Lars is an expert at interpreting these patterns. So the first thing we did, and what you're about to see, are two movies. So one is how Pseudomonas aeruginosa grows over about a 4-day time period. And again, this red stain actually comes from staining of the biofilm. So you're going to see it grow in a time lapse, and then we'll look at the pattern, and then we'll compare that to what happens with our biofilm inhibitor, and then we'll look at images side by side. So the first thing you can see is the bacteria is growing. It starts out kind of white, and then after a certain period of time, you see that it starts to gain red color, and then it starts to wrinkle. And this is when it's forming biofilms. Again, this is just how it will naturally grow. So you can see it has this nice wrinkled phenotype, and that's the end of that video. Okay, so that's just without any of our biofilm inhibitor. So now we add our biofilm inhibitor and watch the same growth. So you can see it kind of starts out the same, it's nice and white. And then this colony, interestingly, becomes a little bit more red more quickly. And you can see that it's wrinkling in a different way. It's actually maybe getting deeper wrinkled and red. So when we look at these side by side, this was unexpected. So if we zoom back out and look at these things, wrinkling and dark red mean you've actually made more biofilm. And so we were like, oh no, our biofilm inhibitor might be making things worse, which was very unexpected compared to what it normally looked like. And when Lars saw this, he was like, this actually really reminds me of this other phenotype that we have studied, which is when you knock these compounds out. So Pseudomonas aeruginosa makes these series of compounds called the phenazines, and they essentially use these like little, Lars described it to me, they're almost like little scuba snorkels for how the bacteria helps oxygenate itself when it's in a really, when it's in these biofilm states. Because you can imagine it's wrinkling to help increase availability to oxygen so it can keep growing to make these deeper wrinkles. And this is basically, since these things can't like pick up and walk, they're using these molecules to essentially do specific kinds of chemistry to allow them to keep living. And so he's like, this phenotype we have here by exogenous treatment of a biofilm inhibitor reminds me of this when we knock all of these out. So again, it's how they're figuring out how to live if they can't make that kind of chemistry.
What's the scale on that?
These colonies, that's a great question. I hit the scale bar. These things are probably like this big. They're like quarter sized, I would say. Yeah.
And do they die off after a while, naturally? Or?
Yeah, if you stop giving them more nutrients, they eventually will die. But Pseudomonas, like if it's on the surface of like a workbench, can persist for up to a couple of weeks. Yeah. Whereas like other microorganisms will die more quickly, but Pseudomonas is really good at like persisting, which is again why it's really problematic in hospitals. So this is really interesting. So we wanted to understand, because we knew that feeding in a biofilm inhibitor was not going to be getting rid of a gene, or in a bacteria, that's not how that works. But we're like, why are we creating that same kind of phenotype? Like what pathway are we activating to show it's as if it's losing these phenazine compounds? So this is where what my lab likes to do comes in. So we wanted to see what was actually happening to these natural products. Because I would argue now, while we like to, find natural products for therapeutic potential, Pseudomonas, like even the bad bacteria, are really good at making natural products. So, not all natural products are good, but so we're like, okay, well, how is Pseudomonas responding to this compound? And this is where the fun comes in. So what I like to do is this thing called mass spectrometry. And the way I like to think about it is it's the world's most expensive balance. So, right, I said earlier, natural products are comprised of carbon, nitrogen, oxygen, hydrogen. And if we look at our periodic table that's conveniently already in this room, what you can see is that every element actually has its own unique atomic weight. So, right, each element has a very different weight. And the interesting thing is, if we think about then these natural products and whether we could weigh them, then they're a compilation of carbon, nitrogen, hydrogen, and oxygen that make these things, then has a very specific mass. And we just like to use the world's most expensive balances to precisely weigh these things. And so again, each of these structures I've shown you are very different as far as like the actual atoms that go into them. And that's what we're exploiting for the measurements that we take. And so again, we can think about these as we know bacteria make a lot of these things. So they're starting out as a really complex mixture. And the important part is I'm in the business of measuring ions. So we either give these positive or negative charges. So right now this would be neutral. So it has no positive or negative value to it. So we turn them into ions, which is essentially just adding, in this case, we'll just say we'll add a proton. And then we can separate them by their size and their masses. So small molecules will, in what we do for time of flight, come out first, separated from really large ones. And ultimately what this ends up being is what I like to call a spectrum. This is the only spectrum I'll show you. But we're dealing with essentially separating things by these masses, and the masses you can calculate based on their elemental formulas. So these really high things, this starts to become like very small protein region, versus over here is, some of these like phenazine molecules that I just showed you from Pseudomonas aeruginosa. So we're just going to be separating based on their mass to charge. when we start out with one single complex mixture. Yes.
So Laura, I've never really fully understood this myself. So how do you put the stuff into the machine? I mean, does it go in as a spray and then you have a charge separating them? I'm not seeing the underlying way that works.
Yeah, that's actually my next slide.
Oh.
So, well, yeah, let's go ahead and get there. So further, the way we like to measure these solid samples is very different. So you're going to be watching this video. So in my lab, we like to do, like, the very technical name is matrix assisted laser desorption ionization. So I'm going to show you how we prep these samples and actually do this. So we have our bacteria, and again, you can envision this is like Pseudomonas just growing happy. We're going to let it grow, and then we physically just pick it up and plop it out. So you'll see the hands in this video are going to put this onto a multi-target plate. So I deal with solid surfaces. And this is the plate that we actually fully introduce into our mass spectrometer, or our very expensive balance. So once it goes in, for the technique that we use, we have to have our analyte of interest, or like these natural products, essentially coated in a matrix. And the matrix is what is going to get our analyte off of a surface and turned into an ion so that we can measure it within the mass spectrometer. So you can see here, this process of adding the matrix is, if anyone has ever sprinkled like powdered sugar over a cake or pancakes, that is exactly what we're doing. So we're using a fine sieve to sprinkle the matrix over our sample. And you can see when it comes out, it's nice and evenly distributed, just like powdered sugar through a sieve over like pancakes. And then there is one little extra trick. So kind of like baking, since we are using lasers, lasers tend to need vacuums. You do not want to put anything wet into a vacuum, it will explode. And I've seen this happen once. So kind of like cooking, what we do is once we have this little wet sample, we actually just take it and plop it into an oven. So it sits in an oven at 37 degrees for 4 hours, and that fully dries the sample down. So once we have a dried flat sample that is coated with this matrix, we put the whole thing into the mass spectrometer. And that is where the fun begins. So we can tell the instrument, measure this entire area at these spatial steps. So we can essentially take data across our entire sample. And what's happening at each data point is we're going to irradiate with the laser. And again, that's going to get our analyte or our natural products off the surface and turned into an ion. And then they're going to be separated just in that tube like I just showed you. So we're going to be separating things based on their masses. And this is going to happen over hundreds of thousands of data points. So we're going to have lots of data in each data point. This kind of becomes a big data problem. But the computer has a solution. So my students like to call this the master spectrum. So rather than looking at every single data point, every single analyte within our data points, we take an overall computational average and then just say, hey, instrument, where was this like pink peak that has a very specific weight in relation to our original biological sample? And so this is what we like to call our hypothesis generating tool. So we're looking at false colored images. So each one of these pixels from where I originally took a data point, I'm coloring based on the intensity of this specific natural product. And so we could say this must be colony associated. This would be colony excreted because we don't see it where that original biological colony was, which is in the center of this. And then something like this is simply the background. And so what we're looking for is how is this chemistry changing when we have treatment and no treatment? And again, since these biofilms grow on surfaces, we're interested in understanding if there was a spatial component to this. Is that all jive? Okay. So, yeah. Do you want? Okay. So, yeah.
The intensity of the color, you said that's intensity.
Yeah. So like how much?
How often is that weight in the material?
Yes. And you can infer that it's also like how much is it at that specific area. So a higher intensity, you could infer it might be like a higher amount of that specific natural product at that specific point.
Is that what you mean by spatial component?
Yes,
Okay.
yes.
Well, what if, you say you're putting something like powdered sugar on there, that must show up on there also.
It
Is that
does.
like a reference material? that you
Yeah.
use?
It can be, yeah. So for us, it can be something that we know, like those are oftentimes the easiest signals to see because we put so much in. You're exactly right, where they end up showing up essentially all over the sample.
Right.
Yeah.
okay.
And so we just ignore those ones. Anything that has essentially like a solid distribution across the whole sample, we just ignore.
I have a silly question.
There's no such thing.
When you say you're ionizing the bacteria, it just occurred to me, is there a possibility of just charging a surface with electricity in a certain way so you can eliminate the separated components? That's a silly question.
Yeah, no, that's not a silly question. So, so this technique was actually invented in the late 80s. And for the very early instruments, they did worry about charging on the surface. And they have changed these in such a way that that's not a problem anymore. But for like experiments that occurred in the 90s, it was absolutely a problem and a consideration of the charge buildup on the surface sample. And it did all sorts of weird things in the instrument. Yeah.
So I need to catch up.
Yeah.
How large are the spots that you're analyzing at each time? Is it-
Yeah, so for us, these are 500 microns for these samples because the Pseudomonas is about as big as a quarter. So we're getting lots of data points across.
500 mic?
Yeah, so 500, I'm trying to think about what that would be. It's pretty small. But, so like a single cell is 10 microns. So it's essentially like the size would be, I guess, 5 human cells is each data point worth of data.
What-
Yeah, I don't know if there's a good,
So
It's like a half a millimeter.
yeah, it's very small, yeah.
for a single molecule, then you're measuring travel time?
Yeah, so it converts the travel time and the time of flight tube to a mass to charge ratio.
Okay. I mean, so it knows when it ionized, and then when that molecule flies to, it goes through some kind of beam and is counted at the end of.
Yep.
Okay.
Yep, that's
Yeah.
exactly right.
So you have a charge pulling it along by charged plates at the end of the tube?
Yes, yeah, so we create, I totally just like finished teaching this in my class, but yes, we manipulate these ions, so like a positive charge, by having a large negative potential to help draw it in. So anytime if we do negative molecules, then we would need positive charges to attract them in. But yeah, that's how we manipulate these things. So if it's not charged, we're never gonna see it. And not everything carries a charge like this. Any other questions? Okay. So I just want to show you a subset of what this data looks like. So here we have our Pseudomonas aeruginosa with no treatment and Pseudomonas aeruginosa with our biofilm inhibitor. And I want to point out 2 signals. So what you're seeing now is a rainbow colored intensity. So instead of single colors, the students I had at that time really liked to show everything in the rainbow inkjet colors, which it's not super colorblind friendly, and I apologize, but it's like too hard to go back and change data from 5 years ago. But this white and pink and red would be high intensity or high concentration of our specific natural product, and then low would be blue and black. So what you're seeing here is when we have our biofilm inhibitor treatment, we see an increase of this specific mass to charge ratio, which turns out to be piochelin. So piochelin is a natural product that Pseudomonas aeruginosa likes to make, and it's very good at chelating iron. So has anyone ever heard like when you're sick, you shouldn't be eating steak? Like you don't want to eat more iron? Yeah, it was kind of like an older, at first I thought it was an old wives' tale, but I actually think there's like truth to this. So these bad bacteria are actually really good. They need iron to live. And this is one of the ways that they get iron out of a system. So it's making more of a compound that's really good at grabbing iron. So that's bad. So we're like, uh-oh. So that was one thing that we observed. And then the other thing we observed were the production of these 2 natural products from Pseudomonas. These are called rhamnolipids. And the important point is these things, again, kind of almost act like soap, and this is how the bacteria moves around more. So again, with no biofilm inhibitor, we see that this is produced on the edge of these colonies. And when we add our biofilm inhibitor, we see that there's a lot more of them around the edge of the colony. So what we interpret this to mean was, it looks like the bacteria is getting better at infecting things, even though it may not be making as many biofilms in liquid. And so the way we wanted to test this was to test what we like to call virulence. So if the bacteria is actually getting worse, even though it may not be able to make biofilms, the only way to test that is to test whether it actually has the ability to damage or kill a host. So virulence is a very technical word, but basically it's the ability to damage host. And these people actually in 2014 had said, if you treated, so their biofilm inhibitor they were trying to bring forward was this like freaky looking compound that's basically an inorganic molecule. I show it here because it's very different than the ones we were working with that we'd isolated from fish as like a, just to say that there are, again, people are trying to bring forward different looking therapeutic entities. And they found in their study using C. elegans, so like baby worm models, that when you give it this biofilm inhibitor, they posited that there's this dispersed cell state and that these were better at killing C. elegans. So again, even though we may be getting rid of biofilms, it looks like we're turning it into something that's even worse for the host. And so this was the only paper I could find at this time that had suggested that this was a phenomena for anyone working on biofilm inhibitors. So what we did was to call, phoned a friend, and we were like, okay, we need to test virulence, and to test virulence, we can't just use bacteria on Petri dish, we have to have a host. So the host we used was the wax moth, Galleria. So these kind of remind me of the little things you might go fishing with, right? Like essentially little mealworms. And the interesting thing about mealworms is in these models, you can tell when they're damaged and dead. So they actually kind of shrivel up and turn black. So this is a very easy assay. So if we treat these and they're getting worse, we're going to get a lot of little shriveled up worms. And so we did this with my friend who's now at Newcastle because he had this model up and running. And the cool thing about this is we can use hundreds of worms to really get a strong readout of whether this is damaging a host. So what I'm about to show you is what we call a Kaplan-Meier survival curve. And what you're going to be looking at is survival on the y-axis. So when 50% of the worms going into the study die, that would be kind of what we consider, that's what we're going to be comparing. And then we're going to be looking at time of the infection model. So if we just give the worms the vehicle or the biofilm inhibitor itself, 100% of the worms survive. So we're seeing both of these lines up here. Over 16 hours, all the worms are totally fine. Great. So the compound is not doing anything. This is, that would be a great drug, right? We don't want it to kill the host. So when we give our worms Pseudomonas, so this is just the abbreviation for Pseudomonas, about half the worms die at about 15 hours. So in a normal model for these worms, Pseudomonas will kill them, and half of them are dead at about 15 hours. Okay, we gave them a biofilm inhibitor. And what we found this time was that the worms died more quickly. So this time in yellow, you can see half the worms are dead now at about 13 hours. And you might be saying, well, Laura, does 2 hours really matter? I would make the argument, if you only had 15 hours to live, you'd sure want those extra 2 hours. So basically what this is saying, and we did this in 100 worms and 3 times. So we did this a bunch, and this kept happening every single time. So what did this tell us? Right, we were hoping to find these biofilm inhibitors. And I think this is why science is really cool, because we helped validate that there was this phenomenon that if you have a certain kind of biofilm inhibitor, you might actually be making the bacteria way worse. So it's even more likely to kill your host. So I think that, again, this is a tool where you know, everyone is looking for these biofilm inhibitors, but at the end of the day, I think you have to be very careful what kind of biofilm, like how you're going about getting rid of the biofilms. Yes.
It would seem like you're exposing the bacteria. If you really wanted to optimize the use of the biofilm inhibitors, you'd add an antibiotic to it
Yeah.
at the same time.
Yes. So we did try that, and you would think that that should work, but I think because we kicked it into this like hyper survival, like kill the host mode, it didn't work like we had hoped. So yeah, I think that was a really great observation, and that this kind of nails in like these natural products, I think, are really valuable for trying to understand what's happening in these systems. Because yeah, if we moved forward with like a mouse model or something, we would have killed all the mice really quickly. So I'm really glad we stopped at worms, you know, to use animals with respect. But again, it was a lot of people are still looking for these biofilm inhibitors, and at the end of the day, now, you know, we can quickly test them to see like, does it increase biochilin and rhamnolipid? Because then I would predict that they would end at the same point our study did. So I hope that I've kind of convinced you that natural products are these really great tools for weeding out and also understanding biology. So yeah, so I kind of called this project New Tricks for Old Dogs, because those bile acids we had known about for a really long time, but we have now used them to discover, you know, and probe biological phenomena. So I think this is where natural products are really cool. And a lot of older natural products have been used to like name things like in our brain, like the opioid receptors. Like opioids are natural products from plants. So I think they're still really valuable because it can help us explain a lot of biology. And so now I want to say, you know, I touched on how this was one story that, again, kind of spanned almost 10 years, but there was some breaks in between. And I want to just add that I think natural products and like the field that I'm currently in, this like mass spectrometry, or the ability to finely measure molecules, they've essentially co-developed over time. So I kind of want to, I thought this would be interesting to talk about. I like to call it the perfect storm. So the field of natural products was, you know, first formed in 1959. Mass spectrometry essentially came online right around the same time. And I used to be on the history committee for our mass spectrometry society. And I learned that I can access all of the old proceedings from that meeting, and found one from 1962 from Klaus Biemann, where he was using natural product or mass spectrometry to study the structure of these organic molecules. And long story short, I pulled this directly from his abstract. And what I want to point out here is that these are two very complicated looking natural products, but what he was able to do was to use these structures to figure out where exactly the difference was between these two. And this is something that I'm going to talk about for what we still do today, even though people didn't realize that this was happening in, I think, when I say 1963. So this is where I kind of want to say what's old is new again, because people knew in 1963 we could do this, and it was essentially kind of rediscovered when I was doing my PhD. So I think it's always important to know, you know, where have we been and where are we going to go? So we know that we can use these, you know, these very precise mass readouts to help figure out how structures are different. So what do we do? How do we do that today? So I just want to say, I kind of walked you through, I kind of skipped to like, what was the fun biology at the end? But I also just want to say, between sample and getting a strain, that there was a whole lot of work. There was probably like a year of my life tied up into like growing this up so I can get enough to figure out what it was so that we could do more. And my old boss actually estimated that it took 355 days, and, you know, including my time and some other factors, that it would have cost $86,000 to have like discovered that bile acid. In today, just adjusting for inflation, that's about $100,000. And you can kind of just imagine that if this is what it takes to discover all of these products, like this is becoming very quickly non-feasible, because it's getting harder and harder to find money to support what we do. And so we want to think about like, how can we lower these costs to do this? And I would also say, I think this is why a lot of drug companies have abandoned natural products. Because the other thing I didn't point out was, you know, this was like We had hoped we were gonna get a biofilm inhibitor, and I got really lucky, but there was no guarantee that we were gonna find that. So it's a little bit of luck to find some of the antibiotics and the anti-cancer agents that we currently have, because it's a lot of really basic science investigation to find these things. And we don't know a priori going into it what we're looking for. So what we're trying to think about now in my lab is like, how do we do this, keeping the cost in mind to make it more efficient? So I'm trying to make sure graduate students don't spend 355 days doing these things, because the faster we can do it, you know, the faster they can graduate and enter into the economy. Anyway, so what do? So again, I kind of want to highlight that natural products is a little bit like luck. So like this is an example of a petri dish where we would have collected sediment from the ocean and just plopped it on to see what would grow. So I just want to know from this room, which one of these would you have picked to enter our like pipeline and why? This is the feel free to.
The fried egg up at the top.
Okay, the fried egg at the top. Anyone else?
Pick the yellow ones, there's more.
Yeah. Oh, like the
The
little ones, like these ones?
Yeah.
Okay. Anyone else?
No idea.
Yeah. So so you could think maybe we could pick them all, but if you have like hundreds of these plates, this just becomes like, where are you going to store these things? How are you actually, again, going to move these through the pipeline? But I also think this highlights that this is so researcher dependent. So there was one student that I worked with, and she liked to pick all the purple ones because she thought they were the prettiest. Right, so that's like totally random luck for like how we find these things. And so what we try to think about then is like, can we do this step using data to inform what we pick? Because we don't want to pick everything, that's not feasible. And again, you also don't want to leave it to like which random researcher was in charge of looking at the plates that day, because they're just going to select, you know, again, it's totally subjective. And so we're trying to remove some of this element of luck. And so again, I told you already how we do the MALDI imaging, but what I didn't tell you was that this MALDI-TOF is actually an FDA approved protocol for identifying bacteria from clinical settings. So like if you go in and they like swab your throat, like they will grow the bacteria and then subject it to a mass spectrometry analysis. And specifically, they look at the protein profiles. So these are abundant in clinical labs, and they're also heavily used in veterinary science. So if you've ever had, you know, your dog or your cat is sick, they will swab them to grow the bacteria, and they identify them this way. Now, this commercial software is a bit pricey. This, again, was a couple years old, so let's inflation adjust to like $50,000 now. But yeah, so this was like, these are in a bunch of clinical labs. And so we're like, okay, this is, and it's FDA approved at essentially making these protein profile fingerprints to identify bacteria. So we were like, okay, what if we do this, but just for natural products now? So we just want environmental bacteria. And then I was like, okay, instead of just proteins, like let's also look at the small molecules because, or the natural products, because that is what we were really good at. And so we did that, and like long story short, it's super simple and it works really well. So we just take these bacteria off these agar plates with a toothpick. Like I am a frequent toothpick buyer from Amazon. We just plop them down, scrape them onto these little stainless steel plates. And then we add a little bit of acid to break the cells up. And then the whole thing goes into the instrument. So again, rather than having our whole flat biological sample, this time we're just using a little bit of that colony that we scraped off with a toothpick. And this is depicting the irradiation event at that specific smear spot of our matrix. You know, matrix is in blue here, our analyte or our natural product of interest is in yellow. So we see that we irradiate these things, they're off the surface, they're turned into ions, and then they're headed down that same time of flight tube. So we like to get the most bang from our buck for the instruments that we do have. So we use them for a lot of different applications. So again, very simple, undergrads do this all the time. And this is an example then of how it worked. So we had a student that took an isolation plate, picked everything off, and then measured the proteins so they could see that indeed, in this case, all the yellow ones were exactly the same thing. And then these two, maybe we'll say egg white only eggs, were different. So these are what we call Pinea bacillus.
And Laura?
Yes.
I wonder if you could just go back that showing how that there, right there.
Yeah.
Okay. Because that looks like I'm still trying to get my head around the basic way your method works. So your laser flashes on that.
Yep.
All these ions jump off the plate.
Yep.
And then they take a different amount of time to be attracted down the other.
Yeah, this happens in like milliseconds.
And the bigger they are, the slower they move?
Yep.
Okay, and so what you get out at the other ends is the ions going through and flip, 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 flip.
Yes.
And that's the core of how this works.
Yeah.
All
Yeah,
right.
that's how they're separated.
All right, and then related to that, so you get that there was a molecule of 11,334.6. From that, what can you say about its structure? I know you guys have
Yeah,
gotten
you're
really
still that-
good about this.
Yeah, you're stealing my thunder. That's in a couple slides. We'll
Oh,
talk about.
okay.
What kind of lasers is it?
So it's a neodymium YAG laser.
So it's used at the 1060 wavelength.
This one is used at 355 nanometers, I believe.
Really? Oh, so it's frequency triple.
What's the sensitivity? Can you say?
Yeah, so that's an interesting point. So interestingly, in this field, we're reliant on the sensitivity will depend on the molecule. So some molecules like to really form these charges. So like I've seen someone quote that it's centimolar, which is like, I can't even fathom what that is. But it's basically something that already had a permanent charge. Yeah, so it'll be natural product dependent. Some of them are much harder to actually turn into ions. Yes.
So the laser itself, that sends ultraviolet, that's the laser ionizes the molecule.
Mhm. Yeah, it's some combination. So this is like the one, I don't like to say chemistry is magic, but this is like legit still magic. So we do not know the order of events here. So we know that we irradiate the sample, but like when exactly they turn into ions, we are not totally sure still. So it's the one area that like, is it that they ionize right after irradiation, or is it there's some sort of chemistry happening in this like plume? Like we don't actually know the order. So yeah, it's the one area I feel comfortable using the word magic still. Yeah.
Does the laser only produce positive ions?
No, that's a great question. That's a great observation. So it will produce neutrals, so things that aren't ionized at all in this plume, negative charges and positive charges. But for how we set these detector plates, so this is set with a negative charge, so we're going to be attracting positive charges towards it, whereas the negative charges that the laser would have created are just repelled.
You're measuring mass to charge ratio.
Yeah.
So if you could get a different amount of ionization of the same kind of molecule, you get multiple signals
Yes.
from that molecule.
Yeah.
Is
Some
that a
people like
problem?
to, no, so some people like to do polarity switching. So like if you can switch positive and negative, then every other shot you're drawing in all your positives, all your negatives. And there is one person, like, I like reviewed this paper, but it was like kind of crazy. So they were trying to do like draw everything in. And I was like, I can't even conceptualize how that would work. But yeah, that's exactly right. So this whole process is inefficient because we're not always creating the same exact charged molecules. Yeah, so like from the amount of sample we have, some of them will be positive, some of them will be negative, and some of them won't be ions at all. And then we can't detect them.
You get +1 and +2 and +3 and +4 for the same molecule.
Yes, that can happen, although it's less likely with this specific ionization.
Okay.
But you get different peaks that you know from the different levels of ionization.
Yes. Okay.
If it's doubly ionized, it's just a different speed.
Yeah, it just changes the Z. Yeah. Okay. So yeah, so we use this to separate the proteins just like the FDA approved method. And then this is the cool part that I think was really impactful for our field, was that we measured the natural products that each of these colonies made, again, from this single smear. And so I want to spend some time walking people through like how we interpret this data. So each of the big circles would be one of these colonies. So 1 is 1, And then each of these smaller nodes would be a specific natural product. So if there was a line connecting the big node to a little node, that was a specific natural product. And so looking at all of these same yellow ones that turned out to be a Rhodococcus species, like, can anyone tell these apart? Not really. Yeah, right. So like each of these, this is basically like I like to call it a tangle. So this indicates that like probably all of these yellow colonies are making very, very similar shared natural products. So if you were going to take one forward for your natural product discovery, you would just pick one of them. We don't need to pick all, how many is that? We don't need to take all 6 of them because there's so much shared chemistry. However, when we look at our 2 egg white omelets, if you will, 7 and 10, what we see here is 7 and 10 do share a lot of chemistry because each of them has a connection to one of these little nodes. So there's a lot of shared natural products, but 7 and 10 also make a bunch of unique chemistry that no one else in this entire plate is making. So what we do then is we would say, we'll probably just take one of the yellow ones and we would take both 7 and 10 because we're trying to maximize is the number of natural products that we can put into our pipeline that might be unique if we're looking for new drugs. Does this kind of make sense? So we're trying to, instead of just randomly picking, we're trying to use data to inform what are we going to spend time looking at, rather than it just being totally random. We're trying to do a data-informed process when because this is the start of every single pipeline these days. So, and as Barry pointed out, this is just a this is like, 117.8. Like, this does not tell us what these compounds are. It's just telling us there's a lot of unique masses or potentially analytes in our sample. And so that is what we're doing now. So we're like, okay, how much further can we identify these things? Or I'm sorry, this is jargon. Dereplication is basically how can we identify the analytes from these samples. So I had a summer student. Oh, and the way, I'm sorry, I was getting ahead of myself. So the way that we like to do this is using a technique called tandem mass spectrometry. So again, you can think of any of your favorite analytes of interest that we've been talking about. It's like any natural product. I actually like to think about each natural product as a gigantic puzzle. And what happens when you throw a puzzle against a wall? Right, it breaks up. So yeah, instead of throwing it against the wall, we actually just apply more energy to it, and we can break the puzzle into its pieces. So we essentially are going to break very specific predictable chemical bonds. And if you're a master puzzler, you don't need to look at the picture to put it back together. Although if you have a picture, then you can also rationalize how the puzzle should be put together. So essentially this process of applying energy to these natural products allows us to get more of the structural information. Because at the end of the day, we want to find new drugs, and we're using this in the smallest way possible to try to get at increasing our chances of finding new drugs. So I had a student do a summer student from Cabrillo College did an exotic freezer expedition. So rather than sending him out to the ocean, I was like, just pull these things out that I'd worked on 10 years ago to come full circle. So he pulled out some of our old fish strains. We've gotten some stuff from a porpoise gut. And Carlos, in about a couple weeks, got all these things growing after they've been in the freezer for 10 years. And applied the process. So like, if I could show that a community college student can do this, then surely anyone can. And so Carlos showed that we are getting, what you're looking at now is a pictorial representation of all the natural products. So each one of these nodes is labeled with a mass. So like 1093. And then it's showing you which of the freezer bacteria it came from. And we're using the fingerprint to say that 1093 is related to 1107. So this is just showing us what kind of relationships exist in the spectra, if there's any at all. So if it doesn't connect to anything, then it might be a unique entity. And so practically, when we took a look at this data, Carlo found known compounds. So these are surfactants that are known to be produced by bacillus, the species of bacillus. So using database searches, he was able to find these are known antibiotic-like compounds. So that's great. So again, just going from a single smear of a colony on a target plate, we're getting the protein information, we're figuring out who has unique chemistry, and then we're trying to get at what is this chemistry all in one shot. So again, these are things just taken off a petri dish that we've regrown out of the freezer in 10 years. So we found known compounds. We were able to database match. These are the iterins. So again, more known compounds. And then I think most excitingly, he found masses for compounds, and we have no idea what these are. So these did not hit, they used to say like a day in the library saved you a month in the lab, like a day on the internet will still save you a month in the lab. So he looked at all of our known databases, and we have no idea what these are, but I think that's the exciting part. So like, this is what we want to figure out now, like, what is this, and does it have any, to restart this process, like, what can we find that it can do for its therapeutic potential? So I hope I've convinced you today that, natural products are really cool, they're really valuable. And right now the name of the game is how do we do this more efficiently? Because I think it's getting harder and harder to do this, and none of this could have been done without a wonderful team of collaborators. So Brian and Ming helped with that bacteria. Alana and Fitnot did a lot of the original biofilm work, and these are the current students that I work with, and we're all dressed as Olympians for Halloween. And of course, none of this would happen without the funding. So I'm happy to take more questions.
All right, good.
That's spectrometry being used on not on bacteria, but on human cells.
Yes.
And can you compare those cells to, say, cancer cells, and can you sort of predict if a cell is going to become cancerous?
That is exactly one other project that we're working on. So we have, I like to call that project Tapping into the Tap, because we're trying to do ovarian cancer detection from cells that are taken in proximity. But yeah, that is absolutely what we're trying to do, because they're so available in clinical labs.
Yeah, so you mentioned the word anti-cancer a couple of times. I was wondering if that's what you're working on also.
Yeah, we work on that as well. Yeah.
I just have a screwball idea. If you took those molecules and accelerated them to a certain potential, so that they're going at a certain velocity, you could put them through a magnetic field.
Yes.
And then they curve and land all the ones with the same M to Z ratio would land in the same place. So you'd have a collection, like a puddle of each different species.
Yep, people do that.
Okay.
Yeah, that is, so they call that like soft landing. So yeah, people are using magnets, and this idea of how we separate things to, some people use that to purify some of the compounds, and also take them on for like more advanced analysis, like cryo-EM. But yeah, that's totally a thing that's come online in the last couple of years. Yeah.
Laura,
Yeah.
So could you say a bit more then about, if you see a molecule of a large molecular weight, you know its weight precisely, how do you go from there to the structure?
Yeah, so a lot of times we'll database search. So like in this case,
Yeah, for those, for example.
yeah, so like these ones, the students actually put that 1107.58
Yeah.
into a database. So they found this one, I can't remember which database this was from, but they found it in a database, and they compared it to, like, this was actually published in that paper. So they matched it to what we had seen. So yeah, so it is possible to search just the masses for databases.
But are they looking just at the mass of the whole product, or of the fragmentation of the fragments?
Both.
Yeah, but the fragments, you have to have the fragments, right?
Yeah.
Okay, you generate,
So maybe related, but do you contemplate using AI to help you identify some of these?
Yeah, that is a great, so I always like to say, like, yeah, so yes. So, yes. So I would say the problem with this is the AI relies on enough metadata. right? So you have to have enough information. So like, this is why I think, protein prediction was, the people that won the Nobel Prize this year was totally built on the backs of everyone that had deposited to the protein data bank, which was a repository that I think is probably 20 or 30 years old. But it was everyone in the field agreed that like, if we're gonna deposit a protein crystal structure, this is the exact data and like, this is the exact deposition that it has to look like. So they were able to train this new protein like AlphaFold, the protein prediction tools, all based on years and years and years of well-collected curated data. So the problem with natural products is like, people are very hesitant, and in a lot of other science fields, people are very hesitant to share their data. And so what we kind of have to balance is like, I'm super excited about these, and a current student is working on trying to solve these structures, but like, just because it's not in a database doesn't mean that it's like not known, right? So yeah, so we're trying to kind of balance like, people have been, so in my lab, like, we're very fastidious depositors in the databases, but I think we don't have enough that's well curated yet. So like, that would be the total dream, because this is still the hard part for what we do. Like that 355 days, like, I would say anywhere from like 2 days all the way up to 6 months could be spent on like figuring out like, what the heck, like, what do I know that I have in my hand? Like, what does it actually look like? Yeah.
I have two related,
Sure.
going back a ways. So you said that they're using this mass spec to identify bacteria.
Yep.
So basically what you're seeing a whole bunch of lines, and it basically acts like a fingerprint.
Yep.
I mean, and I'm just amazed that
That it works.
Yeah. this is-
We do.
Okay, and so related to that, you said it's okay for vets. So is this one of these, it's not sufficiently, has too high an error rate to do for humans?
No, it's used in human and veterinary medicine. It's basically, because before, bacteria could be identified in the clinic by gram staining.
Yeah.
Which is, you're like, okay, what is its shape? Like, how did it stain?
Mhm.
That's not super precise. And then it would take a lot more time or potentially experiments to figure out like, what were you actually infected with? So again, I think this is going from, instead of just looking at what does the bacteria look like, it's like, what chemistry is held within the bacteria, because that's data driven for like trying to figure out what it is.
And it must be relatively inexpensive.
Yeah.
See, you just zap it through, and you get a fingerprint and you put it into a database. No, that's pretty neat.
Yeah, and the instrument on the clinical side, because I have like looked at what it looks like, the software, and it's kind of like red light, yellow light, green light. So if it's green light, it's a positive identification, you move on. And if it's yellow, you still need to do some tests. And then there are still things that are turning red that like, it's, something weird that's not in the database. Or you need to reprep the sample.
So on the next slide that has the sample with all the different colors and the little egg yolks and all that, that all came from just one extraction? Yeah,
Yes.
you were there, or you've gone beyond that, but that particular, I don't even know what it is. Is that a plate?
Yes, that's a petri dish. So this is what we like to grow bacteria on. Let me see.
No, I got that here.
Okay, yeah.
But that just came from one plop of something from the fish guts?
Yep.
Oh, there's a lot going on in there.
Yeah, basically there's this really cool image that I don't have, but you can Google it. Like, if we were to all press our hands onto an agar plate, like, we'd probably all be disgusted by what would grow. But we're essentially always covered, and again, that's like, it's a little both scary and fun to think about. Is like, why aren't we all experiencing infection right now? And I think it's this, there's clearly this interplay of how bacteria learn to live with their hosts. I would say most bacteria don't want us to they want us to be fine. Otherwise they lose their mode of transportation, if you will. So I think, yeah, most bacteria are totally friendly-ish, or are protecting us, much like that original example with the tunicate. That's an example where that bacteria was making a very toxic compound, but the tunicate was cool with it because it protected the tunicate. So I think it's always like two sides of the same coin. Or, we're figuring out how to live in harmony.
Plus, our intestinal system has at least 1,000 different species of bacteria and
Yeah.
fungi. Yeah. Okay, well, thank you very much, Laura. Okay. Okay, so next week, we're going to be looking at viruses and the immune system. All right. Really very interesting talk. Okay.
Can we tell the ones that have biofilms on them up there because of overwriting?
Yeah.
Yeah, I would say that these are probably strong biofilm formers, whereas those ones.
So you haven't